New episode of Zoology, and we will be discussing the perpetuation of life. How does the organelles inside your cell make up tissues, and how these tissues are organized to create organ systems? But how does the life be reproduced and being um, continued all throughout the history of the organism? So this is a lesson which we will be talking about cell division, the cell cycle, and the embryonic development for the animals. So the term cell cycle will refer to how your multicellular animal develops, grows, and maintains and repairs body tissue. So examine yourself when you are wounded. Your body cells will automatically heal the wound on your skin or elsewhere in your body. So that the term itself, the cell cycle, each new cell will receive a complete copy of all the genetic information in the parent cell and the cytoplasmic substances and organelles to carry out hereditary instructions. And these hereditary instructions are being ruled out by the book of your cell, which codes for certain traits and genes and some of the characteristics of the animal. We call it the molecule DNA and the RNA and its kind. Okay, so for the animal cell cycle, being seen in figure 2.3 is divided into different phases. The largest phase or the longest phase, we call it the interphase, which includes the gap 1 synthesis, gap 2, and mitosis. um, these stages are consisting the animal cell cycle, and the cells must faithfully undergo this cycle before they will undergo this mitosis or mitotic phase, which will be including the PMET, the prophase, metaphase, anaphase, and the telophase. So interphase, we'll talk about interphase is the interval between successive cell divisions during which the cell is metabolizing and the chromosomes are directing RNA synthesis. These cells must faithfully passing through the interphase because the metabolism of your cell and the complete requirements that are needed for it to follow the mitosis phase shall be strictly regulated. Okay, so these are the interphase stages. The G1 phase, the first growth phase, metabolism and growth. Second is the S phase, where the DNA is being made, or the synthesis phase. G2 is the second growth phase before it enters mitosis, okay, or karyokinesis. Karyokinesis is the nuclear division. Karyo, which means kernel, in kinesis is division. Okay, so these nuclear parts of your cell will form two daughter nuclei with the same number of chromosomes as the original nucleus. So mitosis will release somatic cells, and they are identical to the parent cells. So this is your animal cell, and the cell cycle. So we have the G1 as G2 phase, and we have the PMAT. And then the last phase of your mitotic phase is the cytokinesis, where the cytoplasm is forming a cleavage furrow. Okay, if you will notice, the cleavage furrow is forming like a cleavage where the cytoplasm contents and subcellular components are dividing and from away from each other. So like the animal cell, the plant cycle also consisting of the same processes. So growth is the increase in cellular mass as a result of metabolism. We see that one in the growing animals, where some animals will start in the fertilization of your egg in the sperm, and then it will grow into a big animal, just like an elephant or a giraffe or a tamarau. So the cellular mass will be aided, okay, by the processes inside the cell. We call it metabolism. Okay, synthesis is the production of DNA and RNA to regulate cell activities. So these are being created from the nucleus, going out of the nucleus to create polypeptide series of proteins and amino acids in the process of translation. Okay, mitosis is the splitting of your nucleus and the equal separation of the chromatids, and cytokinesis is the division of the cytoplasm that accompanies mitosis. So unlike animal cells, plant cells have rigid cell wall, okay, because plant cells tend to have a cell wall. In animals, we don't have cell walls, but we have cell membrane. That's why it does not cleave during the cytokinesis. Instead, a new cell wall is constructed between daughter cells. Okay. Furthermore, many land plants do not have centrioles for the attachment of spindles, unlike the animals which has the centrioles in the middle of their sister chromatids. And these microtubules in these plants form a barrel shape, an astral spindle at each pole. So in your plants, mitosis and cytokinesis occurs basically the same sequence as these processes in your animal cells. So that's the difference between the plant cell and animal cells cell cycle. Okay, so we have asexual reproduction. So some animals aren't using their sex organs to produce. Okay, the production of new individuals by processes that do not involve the gametes, okay, or the sex cells, which occurs in the variety of microorganisms. So we have the fungi, plants, and some animals perform perform this asexual reproduction, or the absence of sexual cells or sex cells. So this is producing an offspring which is which is identical to the parent cell. So the parents and the daughters are the offsprings are identical with one another. So we have different types of your asexual reproduction. This is not dependent, okay, on the presence of other individuals. No egg or sperm is required in asexual reproduction. All the offsprings are genetically identical, except for mutants. Okay. The first type is fission. Okay, we call that subdivision of a cell, or organism, or population, or species, etc., into separate parts. So in bacteria, they perform binary fission because it produces into two separate parts. Multiple fission produces into two separate parts. Okay, will cells, populations, and species, etc. So that's the first type of asexual reproduction. And the second one is sporulation, or multiple fission. So many cells are formed and joined together in a cyst-like structures, just like in protozoans and fungi. Budding, buds develop organisms like the parent and then detach themselves. Okay, so that's common in hydras, some of your fungi like yeast, and certain plants. So they develop like the parent and then detach themselves once they are fully grown. And fragmentation, the last one, is when organism is breaking its body into two or more parts, and each part is capable of becoming a complete organism, just like an algae, your flatworms, and your echinoderms. So we'll talk about that one in the few figures, for example. So this is your sexual reproduction. It's a type of propagation of new organisms through the union of your genetic materials coming from your maternal side and the paternal side. So the sexual reproduction usually involves the fusion of haploid gametes, such as the sperm and your egg. Okay, so these gametes are haploid, which means to say they have only one set of chromosome during a fertilization to form a zygote. So there are some major biological difference between sexual and asexual reproduction. In that way, sexual reproduction produces genetic variation. So the offsprings of your sexual reproduction is varied. Okay, they have different physical features. Whereas the combining of your genetic material from your gametes produce offspring that are different from your parents. So you are different from your parents. And then contain a newer combinations of characteristics. So the term itself is not blending of traits, but rather shuffling of traits. So that's a wrong notion that you are just like a paint. When your mother is like yellow and your father is like blue, then you are green. That's not the case. If you're a green, so if you're color green, if you're a paint, if you refer to yourself as a blending of traits, your siblings must also have the same color too. But no, you're different from your sibling. So this is shuffling of traits. So what traits that are coming from your mother and your father? Let's select this eye color from your dad, and nose length from your mom, hair color from your dad, height from your mom, intelligence from your dad, etc. So that's a shuffling of traits, and these will increase the ability of your species to survive environmental changes and to produce and reproduce in the new habitats. So that's adaptation. Okay, so we will learn that one in the evolution. So the only genetic variation that can arise in asexual reproduction comes from mutations. So this one is a figure of a cinnamon teal, okay, Anas cyanoptera in early spring. So this is a girl and a girl, a male and a female cinnamon teal. Okay. So this one is a type of asexual reproduction with the binary fission. So try to look at this example. So these are for protozoans and other single-celled organisms, including bacteria. So they are dividing into two separate cells. For fragmentation, okay, this is when an organism is breaking into two or more parts, whether it be vertical or horizontal. So you divide them, and then they will be capable of becoming a complete organism when you let them thrive in the wild. So this is a echinoderm and a flatworm. This is an echinoderm. Examples of them are echinoderms and flatworms. So this is a planarian. Okay. So this is a duplicated chromosome which consisting of two identical chromatids at a centrally located constricted centromere. So this is your chromosome. Okay. So we call that chromatid if it's one strand like this one, and then we call them sister chromatids if they are paired, and they are tied together in a centromere. So each of your chromatid is consisting of a chromatin strand. Okay, and these chromatin strand are composed of solenoids. They are wounded with DNA that are wounded with histones that creating solenoids and then creating rosette loops. Okay, and they create the chromatin strand. So that is what we can see inside your nucleus. So they are referred to as the colored bodies because they are stained under the microscope. So they produce high colors, and you can see here in this image is a figure of your mitosis. So the first one is prophase, PMAT. Okay, PMAT, prophase or before. Okay, so this duplicated chromosome consisting of two chromatids which are joined by a centromere. So in this example number 1, we can see asters that are around the centriole. So centrioles are moving on the opposite sides of opposite poles of your cell, and they are joined together. Moved together by the asters. Okay, so they move it on the opposite side of the cell. They will be number 2 is the chromosomes, so the dark stained bodies. The next phase is your metaphase. So they are aligning in an equatorial plane. So they are positioned here, and the spindle fibers from each centriole will then attach to the centromeres. Okay, so number 1 is a aster around the centriole. This red one, these small reds here, are what we call as the spindle fibers, and the chromosomes are found on the either side of your equator. Okay, so the anaphase, the centromeres will split, and then the sister chromatids will separate, and each is pulled on the opposite pole. Okay, so number 1, again, aster around the centriole. Number 2 is spindle fibers, and 3 is your separating chromosome. So next will be your telophase, and the chromosomes will lengthen. Okay, they will become more or less distinct, and the cell membrane forms between the forming two daughter cells. So the nuclear envelope will enclose these materials, and also the cell membrane will enclose the formation of the daughter cells. So number 1 is your new cell membrane, and number 2 is your newly forming nucleus. And the daughter cells are shown in this example, whereas the single chromosomes, okay, former chromatids, so they are called chromatids in your anaphase, but they are now single chromosomes in your newly formed daughter cells. Okay, to continue to lengthen as a nuclear membrane, and then they reform. They try to condense, so the new mitotic phase will occur once more. Okay, so this is cell division is complete. The cell division is complete, and the newly formed cells will grow and mature. So they will undergo the same process in the cell cycle, where entering the interphase and G1 and S and G2 phase, etc. So these are the stages of mitosis in 500X. So what about spermatogenesis? For males and females, they will produce your sex cells, or the gametes. So this is a formation of your sperm cell. We call that one spermatogenesis, producing spermatozoa. Okay, and they are undergoing the same process as mitosis, where the first one is spermatogonium, which is a diploid. Okay, and it undergoes first meiotic division. So for primary spermatocyte, which is a diploid, so this is your primary spermatocyte, they will produce two haploid, okay, spermatocytes, and then these spermatocytes will produce a spermatid. Then it will continuously grow a cap-like structure on their heads and a tail, okay, or a flagellum into the spermatozoans, which are haploid sperm cells. Okay, so these are the parts of your spermatozoa: your head, the midpiece, and the flagellum. So these are unique type of cells because they are moving towards the egg. So they are penetrating the egg for fertilization. So this one is another process we call the oogenesis, which is facilitated also by the process of meiosis, where the oogonium is a diploid, the same thing with spermatogenesis. The primary oocyte is a diploid also. So they will produce a polar body, okay? And then the polar bodies will disintegrate. So we have one, okay, two polar bodies. So on this secondary oocyte, which is a haploid, okay, because the other one is becoming a polar body. So this is fertilized female gamete, okay, with male gamete. So the sperm will contact with the secondary oocyte, and then once they become mature, the zygote will form. So in your egg cells are quite big than the sperm cells. Okay, one of the biggest. The biggest cell in the human body is the egg cell. So they are quite visible to the naked eye. So this one is producing an egg cell. Okay. So this is a figure of your frog testis and frog ovary. So this in a 200X. Okay, the first one is a spermatocyte, number 1, and number 2 is your developing sperm. For frog ovary, we have follicle cells and germinal vesicle and nucleoli. So this is follicle cells are surrounding your nucleoli. Another figure is an intact chicken egg, letter A, and a portion of the shell is being removed to view the internal structures. So these are the structures of your intact your chicken egg. Number 1 is the white substance, or the shell, okay, which is made up of calcium carbonate. Number 2 is vitelline membrane. Number 3 is a yolk. 4 is a shell membrane. 5 is the albumin, or we call that one as the egg white. And 6 is the chalaza, or the dense albumin. And the 7 is the air space. Okay. So now we're trying to see how does the embryonic development of a unfertilized egg, okay, develops. So this is an unfertilized egg. You see it has a nuclear membrane, a nucleus, a nucleolus, and a cell membrane. So this is a typical unfertilized egg. So when it has been fertilized, it will form a fertilization membrane, and then forming a 2-cell stage. So it will be producing 2 different cells, and then it undergoes a 4-cell stage. It becomes 8, it becomes 16, it becomes 32, and it becomes 64. It is multiplied by 2. Okay, so 8 times 2 is 16, times 2 32, times 2 64, and as you go on ahead, it will grow into a this C-star development. Okay, so the first example, we have this blastula. or the blastocoel. Number 1 is the blastocoel for blastula, and then it will form the early gastrula. It has different parts. We have the blastocoel, archenteron, the blastopore, the ectoderm, and the endoderm. So these are important in the embryonic development because these internal structures here will form the internal organs of your animal. So in the late gastrula, we have the ectoderm for number 1, mesenchyme cells, coelomic sac, the mesoderm, the archenteron, and the blastopores. So this example on this image is a bipennaria larva, and a lateral view it shows our oral lobe, the mouth. Here, number 2, number 3 is your coelomic pouch, number 4 is the stomach, and the 5 is the anus. And then it will the sea star will undergo the early brachylaria larva in anterior view, the mouth, the stomach, and the anus. And now you have the new young sea star. So this is just a graphical diagram representing the different stages of the embryonic development of the animals, from fertilized egg until the 16 cell stage. And then this is the frog development of from fertilized egg to early gastrula, shown in diagram and 100x photomicrograph. Okay. So that's the neural tube stage and the longitudinal section. It has formed some of the basic parts, okay, of the organs. To cord for the spinal cord of your animal, of your frog. It has the rhombobencephalon, the mesencephalon, prosencephalon, the oral invagination, which this part will form the head of the anterior end of your animal, the hindgut, the anus, and the yolk, and the liver, and the heart, and the internal organs. So this is the late neural tube stage. It has already the oral plate, the prosencephalon, the heart, the liver, ectoderm, the mesoderm, and the anus. So technically, it grows into a larvae at tadpole stage. So it has the same structures. It has formed the heart, and then the late neural tube stage. It has formed already the eye, the nasal pit, the mouth, and the tail for the mitosis for this animal. And this is a chick embryo development. So we start with the 13th stage, primitive streak, the gastrulation, the neurulation for the 18-hour, 21-hour, the head fold, the neural fold, the muscle plate will form. And then we have the 28-hour stage, the head fold and brain, the artery formation. Most of the muscles plate or the somites will form, and the blood vessels are already can be seen. So the 38-hour stage, optic vesicles, the brain with 5 regions, and the heart. For the 48-hour stage, we have the ear, the brain, the eye, the heart, and the arteries. And the 56-hour stage, we have the ear, the brain, the eye, the heart, and the artery. In the 96-hour stage, we have the eye, the brain, the heart, and the wing. Try to see at the wing. Number 4, the wing has been already formed, this part. And the fecal sac, where it houses the fecal components of your chicken, and the legs are formed in number 6. So these are the major stages of your chick development, and I hope you learned something from our discussion for today. If you have any questions, don't forget to click on the comment sections below. Thank you so much for listening, and have a nice day. Thank you.